We're talking about embryonic transformation.
And we're moving past the simple idea of a fertilized egg. We're going headfirst into this really high-stakes period where a relatively simple, flat sheet of cells.
Just a disc.
Just a disc.
Yeah.
And it turns into a fully organized, segmented, three-dimensional body plan.
It's the ultimate architectural shift. I mean, you think about the stages before this. It was all about getting the materials ready, getting the cells in place.
Right.
But this stage, and our sources really highlight this period around stage 10 to stage 12 of development.
Yeah.
This is about the rapid-fire installation of the whole foundation.
And it's not just building. It's about commitment.
Irreversible commitment. This is the moment, this very narrow window where the cells are basically signing their contracts.
Yeah.
You're going to be bone. You're going to be heart muscle. You are destined to be nerve tissue.
Yeah.
And there's no going back.
Exactly. So our mission for this deep dive is to really conquer the material that covers the start of what's called organogenesis. We need to understand, I think, three core interconnected things.
Okay.
First, how are the three primary axes laid out, the blueprint? Second, the dramatic process of embryonic folding. How do you get from that flat disc to the cylindrical body we all have?
A huge mechanical shift.
And third, and this is maybe the most crucial part, how do all the distinct cell populations, these organized epithelial cells and the migratory mesenchymal cells, how are they partitioned and differentiated into every single foundational tissue in the body?
And, the stakes for understanding this couldn't be higher.
Why is that?
Because if you can grasp the mechanics of this initial commitment process, you really hold the key to understanding all subsequent development. And just as importantly, you understand the origins of all congenital malformations.
It's the root of everything.
It's the root. Why does one specific cell end up forming a piece of your spine while its immediate neighbor travels somewhere else and becomes part of your adrenal gland? The source material we have is it's dense, but it's a detailed map of this commitment. It's like an anatomical GPS.
And our job is to make sure you have the key navigation points locked in.
Yeah.
We're moving beyond just definitions here. We want to understand the mechanism.
The how and the why.
So if we're building a complex body, we have to start with the coordinates. That brings us to our first section, establishing the blueprint. It all starts with these three orthogonal axes. We're talking about the fundamental geometric layout.
We are. We're discussing the three dimensions that define every single vertebrate body. You have the cranial-caudal axis, or C, which is just head to tail.
Head to tail, got it.
Then the dorsiventral, or DV, which is back to belly.
Right.
And finally, the lateral axis, which is simply left-right. All of development hinges on specifying these coordinates first. The sources are really clear that cell populations have to be maintained in specific spatial and temporal arrangements for any of this to work.
It's a prerequisite. You can't start folding until you know which way is up, down, left, and right.
You absolutely can't.
So what's fascinating to me here is the sequencing. Are all these axes established at the same time? Is it just a poof, here's your 3D grid moment?
Not at all. Far from it, actually. The specification of the cranial caudal and the dorsiventral axes happens very early.
Yeah.
It seems to be sequentially specified, and it's foundational.
So they come first.
They come first. In fact, you can see these axes visibly established even before the primitive streak, which is this central organizing structure, even begins to regress.
So the streak itself is part of this process.
It defines the process. The direction that the streak regresses in, the direction it shrinks, that fundamentally defines the primary head-to-tail axis. It's a physical movement that sets the primary orientation.
Okay, so that's head to tail. What about back to belly?
The dorsiventral axis is conferred on the entire embryonic disc very early on as well. It effectively determines the backside versus the front side of what is, at that point, still a flat structure.
Okay, so cranial caudal and dorsiventral are the early commitments. They're almost like hard-coded directions in the system. But then we have the lateral axis, the left-right asymmetry, which you said sounds like a bit of a wild card. Why is that one so different?
That's where it gets really, really complex. The lateral axis develops significantly later, and unlike the other two, which seem to be locked in by these large-scale mechanical movements like streak regression.
Right, physical forces.
Physical forces. The left-right asymmetry has to be actively maintained throughout all the crucial folding stages. It's not a one-and-done deal.
What do you mean by actively maintained?
I mean, it's controlled by differential gene expression. It's controlled by these transient molecular cascades. It's not just a consequence of the physical folding of the sheet of cells. It requires active, ongoing molecular mechanisms to suppress left-sided characteristics on the right side of the body, and vice versa.
So it's a constant balancing
It's
act.
a very delicate one.
This sounds like a lot of responsibility for what is, at this point, still just a flat disk of cells. And this all comes from the inner cell mass, the ICM, right? That's the source of all these embryonic structures. How does the ICM even set up these initial coordinates before the folding kicks off?
The ICM is absolutely where the process begins. It separates from the outer tissue, the trophoblast, which goes on to form the placenta.
Right.
That separation and the subsequent formation of the embryonic disc is the very first step in positioning the body plan relative to all the extra embryonic tissues. The initial commitment of the cells within that disc, the epiblast cells, that's what defines the orientation of the primitive streak. And once that orientation is locked in, bam, the CC and DV axes are set.
But here's the kicker you mentioned.
Here's the kicker. The flat disc is, by its nature, two-dimensional. It's only when the cells commit to this pre-programmed axis layout that the spectacular transformation, the folding, can finally begin. That's what turns the 2D map into a 3D structure.
Which brings us directly to that great architectural maneuver, embryonic folding. The sources describe this process where the flat disk is transformed into a cylinder, and it happens between stage 10 and stage 12. This is what defines the entire dorsal and ventral aspects of the developing embryo.
It is the great transformation.
So let's start with the cranial caudal folding. The description sounds like the head is literally tucking itself under.
That's the perfect visualization.
Yeah.
It really is. Cranial caudal folding involves the cranial, or the head end, of the embryo folding sharply ventrally so, bellyward toward the caudal, or tail end.
Can you give us an analogy for that?
Imagine you have a sheet of paper, and you put a very heavy object on one end. That end is going to curl under and inward on itself. That's essentially what's happening to the head region of the embryo.
And the consequences of this rolling motion are profoundly reorganizing, right? It's not just changing the shape, it's physically relocating major future organs.
Precisely. The consequences are massive. This movement incorporates the cranial endoderm, the inner layer, and pulls it centrally to form the foundational structure of the foregut.
The start of the digestive tube.
The very beginning. But the most dramatic relocation by far involves the heart.
Okay, tell me about this.
Before this fold happens, the region that will become the heart, the cardiac primordia and the future pericardial cavity where the heart will sit, are located very, very far cranially. They're almost in front of where the head is going to be.
So the heart starts out in front of the brain.
In a manner of speaking, yes. The tucking movement physically repositions this whole proamniotic and pericardial region ventrally. It causes the cardiac primordia and the major veins that will return blood to the heart to shift into their definitive thoracic, their chest location.
So the heart is literally moved into its chest cavity position by this head-to-tail fold. That's an enormous physical feat, just accomplished by cells migrating and a sheet of tissue moving. What about the nervous system? Does this affect it?
Oh, it has a massive impact on the neural structures. As the cranial end folds under, the midbrain region is projected forward and becomes distinct. This physical projection is the first visibly clear evidence of the brain forming its distinct future anatomical regions.
Wow.
This shift of the neural plate and all the associated structures establishes the fundamental organizational axis of the entire central nervous system.
So if the cranial caudal folding is the tuck, then the lateral folding must be the necessary follow-up. You called it the hugging or zippering motion.
The hugging is essential for closure. You can't have a tube without closing the sides. So lateral folding involves the lateral plate mesoderm folding ventrally on both sides of the embryo. This runs concurrently at about the same time between stage 10 and 11.
And what does that convergence achieve structurally? What's the point of zippering up the sides?
It achieves the classic tube-within-a-tube architecture that defines all vertebrates. First, it brings the bilateral structures, the things that come in pairs, like the body wall and the developing limbs, into their appropriate lateral positions. By folding the sides of the embryonic disc into the body, it transforms that simple endoderm sheet into the fully enclosed tripartite gut tube.
Okay, so that's where the foregut, midgut, and hindgut come from.
Exactly, it defines them. And second, and this is critical, this ventral folding forces the intraembryonic colome, which is the future body cavity, to form by sealing the space between the layers of that lateral plate mesoderm.
So let's connect this back for the listener. What does this all mean for you? These two folds, the tucking and the hugging, they're the crucial architectural phase. They transform the flat map, that two-dimensional disc, into the functional, segmented, three-dimensional tube structure of the body.
And once that tube is formed and closed, everything else is locked in. The organization of all your internal organs, the placement of your limbs, the basic layout of your digestive tract, it's all irrevocably set.
And this entire morphogenetic shift is complete in just a few days.
It's breathtakingly fast.
So we mentioned earlier that the left-right axis is the last one to develop, and that it needs this active maintenance. It seems so vulnerable compared to the solid early commitment of the head-to-tail axis.
It is a profound difference, and it really touches on the robustness of the whole system. While the CC axis involves this large-scale physical movement, the lateral axis is fundamentally about differential gene expression. It's about active signaling cascades that are happening transiently, often centered around a specific region called the node in the early embryo.
So it's chemistry, not physics.
It's almost entirely chemistry. It requires constant active molecular mechanisms to ensure that the left side develops differently from the right.
So let's delve into this molecular babysitting that's required. What molecules are we talking about here? What's involved in this differential gene expression?
Well, the sources indicate that some of the key players are molecules with names like nodal and lefty.
Nodal and lefty.
Nodal is a signaling molecule, a protein, that tends to be restricted to the left side of the embryo. It often sets up the initial signaling pathway for leftness.
Okay, so nodal says this is the left side.
In a sense, yes. And then lefty acts as an inhibitor. Its job is to prevent nodal signaling from spreading over to the right side. So you have this dynamic balance, an activator, nodal, that's restricted by an inhibitor, lefty, and that's what maintains the asymmetry. If that balance is disrupted, you get defects.
And why is this distinction between the sequential specification of head to tail and the actively maintained asymmetry of left-right? Why is that so significant clinically?
It raises a really important question about robustness versus flexibility. Defects in this delicately maintained lateral asymmetry lead directly to conditions like cetus inversus.
That's where all the organs are flipped, right? A mirror image.
Exactly, a perfect mirror image.
Mirror.
Or, more severely, you can get cetus ambiguous or heterotaxy syndromes where the organs are randomized or just completely disorganized.
That sounds much more dangerous.
It is, because the heart is the most dramatically asymmetric organ we have. So these defects often manifest as major, life-threatening cardiac or visceral problems. It tells us that while the main body axis is stabilized early, this left-right axis is a late-developing, very complex addition to the vertebrate body plan that carries significant risk if that molecular signaling fails.
The sources draw a comparison between this kind of developmental regulation in vertebrates and what we see in invertebrates, like the fruit fly, Drosophila. What does that comparison tell us about the evolutionary jump we're looking at here?
It tells us that while some general developmental pathways for building an animal body, things like axial patterning, for instance, might be conserved across different phyla, specific systems like left-right determination are relatively unique to the vertebrate lineage.
Why is that?
Well, vertebrates require this very high degree of internal organ arrangement. Your heart has to be on the left, your spleen on the left, your liver mostly on the right. Invertebrates just don't have that level of internal complexity.
So this specificity highlights the evolutionary complexity that was added to the vertebrate body plan.
Absolutely. It relies on transient embryonic structures and specific gene interactions that just aren't replicated in the insect world. It shows us that the tube structure we built through folding required a highly sophisticated active system to manage the asymmetry of everything inside it.
So if the axes are the blueprint and the folding is the structure, now we have to move into the materials, specialized cell populations to actually build the walls, the pipes, and all the internal machinery.
Yes, and to do that, we must begin with the foundational divide in all of embryogenesis. It's a concept that underpins everything we're about to discuss, the distinction between epithelial and mesenchymal cells.
The two basic states of cells.
These two states are the entire vocabulary the embryo uses to construct every single tissue.
Okay, so how do we define these two basic opposing building blocks? Let's start with epithelial cells.
Epithelial cells are the organized settlers. They're the builders. They're characterized by strong polarity.
Meaning they have a top and a bottom.
A defined apical, or top surface, and a basal, or bottom surface. And they are locked together by very specific adhesion complexes. The sources even mention some of the defining proteins that establish this organization, with names like crumbs and scribble.
Crumbs and scribble, I love that.
It gives you a sense of the structural nature of it. Epithelial cells organize into sheets or tubes or boundary layers. Think of your skin or the lining of your gut. They are inherently structural. They provide barriers and conduits.
They are the organizational boundary setters. So what about the mesenchymal cells?
They are the dynamic movers. the pioneers. They were loosely packed, highly migratory, and they lack that strong cell-to-cell adhesion. Their strength lies in what they produce,
Yeah.
the extracellular matrix, or ECM,
And they form things like bone, cartilage, blood, muscle.
all the connective tissues. The sources stress that development isn't static. It's this constant process of movement and rearrangement. And a key event we absolutely must understand is the epithelial to mesenchymal transition, or EMT.
Okay, let's spend a moment on EMT because this seems central to how some of the most critical tissues we're going to talk about, like the neural crest and the skeleton, are even formed. What's the mechanism behind this transition?
EMT is the process by which an organized, stationary epithelial cell loses its strong connections to its neighbors and acquires the ability to migrate.
It decides to leave home.
It packs its bags and hits the road. At the molecular level, this is often triggered by the loss of specific adhesion molecules, primarily a protein called E-cadherin.
So it's like unzipping the Velcro between cells?
That's a great way to put it. When E-cadherin is suppressed or degraded, the cells detach from their neighbors, they change their shape, they become modal. This allows them to break free from the sheet, travel sometimes very long distances through that mesenchymal matrix, and then often redifferentiate into a completely new structure in a new location.
So it's the molecular switch that allows for complex migration and all the internal differentiation to occur.
It's the key to building a complex three-dimensional organism from simple sheets.
Okay, with that in mind, let's start with the deepest layer, the endoderm. This layer lines all the internal structures. It originates from the cranial end of the primitive streak, and the folding we just discussed is what makes this layer functional.
Absolutely. As the embryo folds, the endoderm is incorporated. It folds to create the stomodium, the primitive mouth at the head end, and the proctodium, the primitive anus, at the tail end.
And that folding action is key because it transforms that flat sheet of endoderm into the fully enclosed tripartite gut tube, the foregut, the midgut, and the hindgut.
And what's astounding is how this simple lining then buzzes out to create pretty much all of the secretory and absorptive machinery of the entire body.
Okay, let's unpack the foregut first.
Yeah.
This sounds like the big producer of organs for the upper body and the respiratory tract.
The foregut is a powerhouse. It gives rise to the entire pharynx and all the epithelial, secretory, and duct cells of the respiratory tract, so the lungs, the trachea, and the esophagus.
Man for the digestive path.
Moving further down, it forms the epithelial lining and all the associated secretory and duct cells of the stomach, the liver, and the pancreas. It also forms the general follicular cells of the thyroid gland. It is an extremely dense region of organ commitment.
But the sources specify even more complexity, especially with the glands that come from this region.
Yes, the complexity here is immense. The endoderm that forms the epithelium of the pharyngeal pouches gives rise to highly specialized structures that are crucial for your immune and endocrine function.
Such as?
This includes the thymus, which is fundamental for developing your T cells, your immune cells. It includes the parathyroid glands, from pouches 3 and 4 specifically, which regulate your body's calcium levels. And it includes the calcitonin-producing C cells of the thyroid.
So this one small region of a seemingly simple epithelial sheet gives rise to major system-wide regulators.
It's a perfect example of localized specialization.
Okay, moving down to the midgut. This sounds like the dedicated workhorse of the digestive tract.
It really is. It's focused entirely on processing and absorption. The midgut endoderm forms the epithelial, glandular, and duct-lining cells of the majority of your small intestine, your appendix, the cecum, and the ascending and transverse parts of your colon.
It's the central absorptive machinery.
Designed for high-efficiency nutrient uptake. That's its whole job.
And finally, the hindgut, which manages the exit and the reservoirs.
The hindgut forms the epithelial lining, the glands, and the ducts of the descending colon, the sigmoid colon, and the rectum, the final stages of the digestive tract. But crucially, it also forms the epithelial lining of the urinary bladder and the urethra.
And these distinct regions for gut, midgut, hindgut, they aren't arbitrary.
Not at all. They're defined by the physical process of folding, and then subsequently they're controlled by highly regional gene expression patterns that ensure the correct organs develop in the correct order along the tube.
All right, now let's flip to the outside, to the ectoderm. This layer differentiates into our protective outer layer, the surface ectoderm, and the foundation of our entire nervous system, the neural plate ectoderm. Let's start with the surface, the skin and sensory layer.
The surface ectoderm is our outer boundary. It's our initial point of contact with the world. So it has to be a highly protective and sensitive layer.
What are the key sensory structures that are derived from this outer sheet?
This is a critical list to understand. From the surface ectoderm, we get the olfactory receptor cells and the olfactory epithelium for our sense of smell. We get the specialized structure of the lens of the eye, which is an important distinction we'll come back to.
Okay.
And we get major parts of the ear, specifically the otocyst, which becomes the inner ear, and the internal layer of the tympanic membrane, or eardrum.
And in terms of protection and barriers, what does it form?
It forms the entire epidermis, the outermost layer of our skin, along with all of its appendages. So hair follicles, sebaceous glands, sweat glands, plus the epithelial lining of your oral cavity and the external acoustic meatus, your ear canal.
The sources mention something specific about the basal lamina here.
They do. They mention the enduring nature of the basal lamina of the epidermis, which forms this continuous physical foundation throughout your entire life. It really highlights the permanence of these early epithelial layers. They set a boundary that lasts.
Okay, moving inward now, let's discuss the neural plate ectoderm. This is the commitment to the central nervous system, the CNS. And this process, primary neurulation, is happening right around stage 10.
Primary neurulation is the formation and the closure of the neural tube. It's a massive event. The neural plate forms a groove, and the lateral edges of that groove fold up and fuse at the midline, creating a tube that completely sinks beneath the surface ectoderm.
So the skin zips up over the top of it.
Exactly. This single massive morphogenetic event defines the structure of the entire CNS.
And that neural tube is the source of all the gray and white matter of our brain and spine.
It's everything. It becomes the entire brain and spinal cord, including all the major divisions we recognize in the adult, the cerebral hemispheres, the midbrain, the cerebellum, the pons, the medulla oblongata.
And the supporting cast.
Crucially, it also gives rise to the inner lining of the ventricles in the brain, which is called the ependema, and the primary supporting cells of the CNS, the astrocytes, and the myelin-producing oligodendrocytes.
This also brings us back to the sense of sight where you said there was a split origin we needed to remember.
This is the key distinction. The surface ectoderm formed the lens.
Right.
But the retinal tissue and the optic nerve, cranial nerve 2, are direct outgrowths and derivatives of the neural plate ectoderm.
So the retina is actually part of the brain.
Anatomically and embryologically, it's considered a direct extension of the brain, not just a simple external sensor. This also includes the epithelium of the iris, the ciliary body, and some of the corneal processes.
Okay, that covers two major epithelial populations. Now let's look at the third one, the coelomic epithelium. This sounds like the specially structural lining of our internal body cavities, right?
It is the inner architectural lining. It ensures that the entire visceral structure is encased and can move without friction. It's derived from the lateral plate mesenchyme after that mesenchyme splits, forming the parietal pericardium and the peritoneal column.
And its main job is to form the serous membranes,
which are these incredibly smooth, protective linings that reduce friction as our internal organs move. You know, your heart beats, your lungs expand, your gut churns. They need a slippery surface to move against.
What specific membranes are we talking about here?
We're talking about the thin, slippery layers of both the visceral and parietal layers of the pericardium, which surrounds the heart,
Okay.
the pleura, which surrounds the lungs and lines the thoracic wall, and the peritoneum, which lines the entire abdominal cavity and covers the organs within it.
So it's basically the biological equivalent of lubricating oil.
A very sophisticated one, yes. This epithelium also plays a vital role in forming internal dividers, like the septa and mesentery, including a structure called the septum transversum, which is critical for separating the thoracic and abdominal cavities.
So it provides the essential boundaries and the necessary slipperiness inside the body, but the sources highlight a really profound non-structural role too, specifically in the urogenital system.
This is perhaps its most specialized and surprising role.
Yeah.
The coelomic epithelium gives rise to the epithelial lining of the urogenital ducts, the mesothelium of the tunica vaginalis, and the epithelial lining of the suprarenal gland cortex. But most importantly,
Yes.
it forms the germinal epithelium of the gonads.
Germinal epithelium is a powerful term. But wait, I thought the germ cells themselves, the precursors to sperm and eggs, migrate from the yolk sac. So what exactly does this epithelium contribute to reproduction?
It contributes to crucial support cells. It builds the architectural foundation of the gonads. So in the female, this epithelium gives rise to the follicular cells that surround, protect, and nourish the developing oocytes.
And in the male?
In the male, it provides the Sertoli cells, which are absolutely essential for nourishing and regulating the process of spermatogenesis.
So this is a remarkable demonstration of how this one epithelial layer can shift its function from simple friction reduction, like in the peritoneum, to highly complex endocrine and reproductive support in the gonads.
It's a fantastic example of developmental versatility.
Okay, we've established the epithelial populations, the boundaries, tubes, and linings. Now we move into the middle layer, the mesenchyme. This is the muscle, bone, blood, and connective tissue. This is the source of our motion and our structure.
And mesenchyme, as we said, is our early critical population of migratory, loosely packed cells. And its defining feature is its ability to produce and interact with its own extracellular matrix, the ECM.
So why is the ECM so vital? Why do we need to spend time on it? It sounds a bit like just the intercellular filling, the packing peanuts between the important parts.
It is so much more than filler. It is the scaffolding, the signaling hub, and the migratory highway map, all rolled into one. The ECM regulates cell interactions, adhesion, growth, and most importantly, it provides directional cues for all those newly migratory mesenchymal cells.
So it's the construction site where every beam and every road sign dictates what happens next.
That's a perfect analogy.
What are the key components of this construction site? What is the ECM actually made of?
It's a complex soup. Key components include large glycoproteins, proteoglycans, and a complex network of collagens like the highly structural types I, 3, and V. And a protein called fibronectin is particularly important here.
What does fibronectin do?
It acts as an adhesive molecule that essentially paints the migration paths for cells. So when a cell undergoes that EMT we talked about, it interacts with the specific fibronectin patterns in the ECM, and that tells it which direction to migrate in and, in some cases, what to differentiate into when it arrives at its destination.
Okay, so with the ECM as the roadmap, let's unpack the 4 primary populations of mesenchyme that appear early on. They migrate laterally from the primitive streak area and define this whole middle layer.
We distinguish them based on their physical arrangement along that head-to-tail axis. First, you have the axial mesenchyme, which is the core rod. Then, moving outwards, the paraxial mesenchyme, the segmented blocks. Then the intermediate mesenchyme, which will become the urinary system. And finally, the lateral plate mesenchyme, which forms the body wall and the organs. This physical arrangement directly reflects their fundamental functional commitment.
Let's start at the center then. The axial mesenchyme, the core rod. This originates from epiblast cells migrating through the primitive streak to the very cranial end, forming the precordial plate and the notochordal plate.
And the notochord itself is functionally monumental, even though it's a transient structure.
It doesn't stick around.
It doesn't. But while it's there, it's the core rod that sends out these powerful inductive signals that influence the overlying neural tube and pattern the underlying endoderm. Without the notochord, the structure of the CNS would completely collapse. The sources even confirm that the notochord is surrounded by its own basal lamina, which helps it maintain internal pressure and apply that signaling pressure to the surrounding cells.
So it's primarily a signaling center. What specific signals does it send out to dictate the development of its neighbors?
The most famous signal it produces is a protein called sonic hedgehog.
Sonic hedgehog, of course.
The notochord is the major ventral source of sonic hedgehog, or shh. This signal diffuses dorsally, upwards, and it instructs the cells of the overlying neural tube to differentiate into ventral structures, like the floor plate and all of the motor neurons.
And does it signal downwards too?
It does. It also patterns the adjacent paraxial mesenchyme, instructing the cells that are closest to it to make that transition into becoming skeletal precursors.
So it's less about the structure it becomes and much more about the command center it provides. What happens to it eventually? Where does it go?
Its signaling job is done relatively early in development. So while it's transient, its fate in adults is to be primarily replaced by the vertebral column. The bones of your spine grow around and obliterate most of it.
But not all of it.
Not all of it. Residual notochordal cells form the gelatinous center of our intervertebral discs, the nucleus pulposus. So its temporary existence is essential for patterning the entire embryo, but its structural contribution in the adult is minor compared to its neighbors.
Okay, moving out from the axis, we get to the paraxial mesenchyme. This is where segmentation begins, defining the repetitive structures of our trunk. This mesenchyme forms the blocks known as somites.
Exactly. The cells forming the paraxial mesenchyme migrate just lateral to the axial mesenchyme, and they condense into these epithelial blocks called somites. This segmentation, which appears first in the trunk region, is the body's original system of organization. It provides the segmental basis for the spine, the muscles, and the skin.
And the differentiation process within these somites is rapid and highly specific.
Very. Somites are the segmented template.
So let's detail the three main parts that a somite differentiates into, because this seems to be the source of all our voluntary movement and much of our skeleton.
The key here is that the epithelial block of the somite immediately begins to tear itself apart using EMT to fulfill its different functions.
Okay, what's first?
First, the sclerotome. This is the part of the somite that's nearest to the notochord in the neural tube. These cells undergo a major epithelial to mesenchymal transition.
There's EMT again.
It's everywhere.
Yeah.
They break away from the block and they migrate medially toward the center. Their job is to form the vertebrae and portions of the ribs. This EMT is essential because they need to be mobile to surround and replace the notochord.
So the skeleton is literally migrating into place, segment by segment. That's the skeletal segmentation locked in. What about muscle and skin?
That comes from the dorsal portion of the somite, a structure called the dermomyotome. And as its name suggests, this structure divides further.
Into dermis and myotome.
Precisely. The dermis portion forms the dermis of the skin, but only over the dorsal regions of the body, so your back. The myotome portion forms all voluntary muscles, all skeletal muscle of the back, the trunk, and importantly, the limbs.
That is an enormous commitment. The paraxial mesenchyme, via the somites, is the source of the entire voluntary skeletal muscle system, plus the axial skeleton.
Yeah.
Let me ask you this. What's the specific advantage of forming the somite as an epithelial block first, before it just splits apart?
That's a great question. That initial, rigidly organized epithelial form helps to establish clear, segment-specific boundaries. It ensures that the specific gene expression patterns for, say, thoracic segment 10 don't just spill over and get mixed up with segment 11.
So it's like building with discrete Lego blocks.
Exactly. It's only once those boundaries are firmly established that the internal cells can be released through EMT to migrate and form structures like bone. This ensures that each vertebra and each muscle segment is distinct and correctly aligned with the nervous system.
Next up, nestled between the segmenting somites and the expansive lateral plate, we find the intermediate mesenchyme. This is a much smaller population, but critically specialized.
The intermediate mesenchyme occupies that narrow strip between the paraxial and lateral plate mesenchyme, and it commits entirely to forming the urogenital system, the machinery for fluid balance and for reproduction.
What are the key derivatives here? How does it break down the functional kidney components from the ducts?
Well, it gives rise to both the epithelial and the mesenchymal parts of the gonads, working alongside the coalomic epithelium we already discussed. But its primary role is building the kidney. It forms the specialized filtration units, the nephrons, and the internal collecting ducts.
And the plumbing. Yeah.
It also gives rise to the entire excretory duct system, specifically the Müllerian and Wolffian ducts, which are the precursors of the male and female internal reproductive tracts.
So it's the manager of filtration and the architect of our reproductive pathways. A small but deeply critical piece of the puzzle, and it's positioned strategically between the segmental muscle and bone on one side and the visceral linings on the other.
Its position is everything.
Now for the biggest player, the lateral plate mesenchyme. This is the most expansive mesenchyme, forming the rest of the body structure. And it's defined by its split into two layers, separated by that intraembryonic column.
A division that was driven by the lateral folding we talked about right at the start.
Let's look at those two layers, starting with the outer one, the one associated with the skin.
That's the somatopleuric mesenchyme, also called the parietal layer. It's associated with the surface ectoderm, and it forms the structures of the body wall and the limbs.
What are its most extensive derivatives?
The somatopleuric layer forms the dermis and the connective tissue of the ventral body wall, so the front and sides of our body. It provides the connective tissue and the skeletal support for the limb buds.
But not the muscle.
Excluding the muscles. Remember, those come from the somites, migrating out into the limb buds. But here's an incredible fact from the sources. It also contributes to a highly specific migratory cell population within the CNS.
What? A cell from the body wall ends up in the brain?
Yes. The microglial cells, the resident immune cells of the brain, originate from this layer. Think about that span. From limb bones to brain immunity, all from the same initial sheet of cells.
That is a massive span. Okay, now for the inner layer. The splanchnopluric mesenchyme or the visceral layer, which is associated with the endoderm?
Splanchnopluric layer is responsible for forming the structural support of all the internal organs. It's the hidden machinery that allows them to move and function.
So this means the muscle and connective tissue of the gut and lungs.
Exactly. It forms the smooth muscle and the connective tissue of the respiratory digestive tracts. It forms all the viscera, everything except that inner epithelial lining which came from the endoderm.
Yeah.
It also contributes to the smooth muscle lining of our blood vessels. But most remarkably, the cardiac muscle, the myocardium of the heart, is formed from the angioplastic mesenchyme, derived right here.
So if you trace your heart tissue, the inner lining, the endothelium, and the outer lining, the pericardium, are derived differently, but the functional pumping muscle of the heart comes from this splanchnopleuric mesenchyme.
And that interaction, the endoderm lining collaborating with the splanchnopleuric mesenchymal support, that is the foundational organizational strategy of virtually every single internal organ.
We have to dedicate some time to the angioplastic mesenchyme specifically, the system that will build the entire circulatory network and the blood itself.
It's the life support system.
So where exactly do these crucial cells originate within that splanchnopleuric mesenchyme?
The sources outline a complex dual origin. Cells that are derived from the splanchnopleuric mesenchyme in the trunk region, in these specific focal points, they become the precursors for the entire circulatory system. These cells differentiate into endothelial cells, which form the first embryonic vessels, including the endothelial lining of the heart.
And the blood cells themselves, where do they come from?
The sources are very careful to distinguish the very first origins. The earliest blood cells actually derive from extraembryonic mesenchyme, specifically from the yolk sac and a structure called the allantois.
So from outside the embryo proper.
Right. However, the definitive long-term fetal blood system arises from intraembryonic sources. The first hematopoietic, or blood stem cells, originate in the splanchnopleuric mesenchyme of the trunk region. So it's a transition from a temporary external blood supply to the permanent internal system.
Okay, let's connect this back to function. The development of this vascular network isn't just about cells differentiating, it's about patterning, right? Building a network.
It is a dynamic patterning process. Vessel formation involves two main methods. There's vasculogenesis, where endothelial cells differentiate in situ, right where they are, and form brand new vessels. And there's angiogenesis, where new vessels bud, sprout, and branch from pre-existing ones.
And this whole process is tightly regulated.
Tightly. The surrounding non-angiogenic mesenchyme sends out signals that determine where blood flow is needed and how the network should be routed across the rapidly growing body plan.
Okay, that covers the 3 germ layers. Now let's turn to section 4, the neural story, central, peripheral, and the highly migratory cells that connect them all.
The command and control systems.
We've established the CNS begins with the neural plate ectoderm forming the neural tube around stage 10. Let's dive deeper into how that tube becomes segmented, especially in the head region where there are no somites.
Right. While the trunk relies on somites for its segmental organization, the head relies on segments called rhombomeres. This segmentation of the hindbrain into these blocks R1 through R7 is visible around stage 14, and it coincides with the development of the pharyngeal arches.
Why are these rhombomeres so crucial for the head? What do they organize?
They are the organizational structure that patterns the branchial or pharyngeal arches. Each rhombomere is a specific genetically defined developmental compartment. They determine which specific cranial motor nerves emerge and where they project.
Can you give an example?
Sure. Rhombomeres 1 and 2 project to the first pharyngeal arch, which will become your jaw structures. Rhombomere 4 project to the second arch, which develops into the hyoid bone and your middle ear structures.
So the rhombomeres define the motor control for the entire face and throat. And the sources also mention a unique feature in human embryos regarding segmentation here.
Yes. The somites, which define the trunk segmentation, are only present rostral so, headward to the 4th somite in the human embryo. This means the head has to rely almost exclusively on the segmented rhombomeres for its structural and neurological organization.
Which highlights a specialized evolutionary pathway for how our heads develop, distinct from the trunk's socomite-driven system.
Absolutely.
Okay, here's where we meet the absolute rock star of embryogenesis, the neural crest. It's often called the 4th germ layer because of its incredible, unmatched versatility.
It truly is a unique population of cells. The neural crest arises from the boundary between the neural plate and the surface ectoderm. It's the region that zips up to form the neural tube. These cells form around stage 9 and 10, and their commitment to migration is immediate.
And their power comes from that defining feature we keep coming back to, the epithelial to mesenchymal transition.
Exactly. They are perhaps the most famous example of EMT in the entire body. These cells must break away from that organized epithelial sheet using molecular cues like the suppression of E-cadherin. They become loosely packed and migratory, and then they travel throughout the entire body to form an astonishing number of different structures, spanning all three primary germ layers in terms of function.
And the migration paths are separated into head neural crest and trunk neural crest, each with very distinct fates. Let's start with the head.
The head neural crest migrates around the closing neural tube to populate the pharyngeal arches through 6. Because they are situated in the head, they contribute to much of the non-neuronal structure there, a function that, in the trunk, the mesenchyme usually handles.
So what specifically do these migratory head cells build?
It's an incredible list. They form the sensory ganglia of many of the cranial nerves. They form the bones, the cartilage, and the connective tissues of the entire face and neck.
Yeah.
That includes your jaw, the tiny middle ear ossicles, and even the dentine of your teeth.
And then the heart connection.
They're also crucial for forming the connective tissue of the heart, particularly the septation of the outflow tract, which ensures the aorta and the pulmonary artery separate correctly. This is why if head neural crest migration fails, you get severe craniofacial and cardiac defects at the same time.
They literally build the entire bony structure of the face. That's profound. Now, for the trunk neural crest, which forms caudal to the hindbrain, below the head.
The trunk neural crest follows 2 major migratory routes. The first is the dorsal route.
Going towards the back.
Right. This is the superficial route moving just beneath the surface ectoderm. These cells differentiate exclusively into melanocytes, the pigment-producing cells in your epidermis and hair follicles.
To your skin and hair color. And the second route.
The second is the ventral route. This is the deep route. These cells travel through the somites and down into the viscera.
And what do they become?
They form the entire peripheral nervous system outside of the CNS. The sensory ganglia, like the dorsal root ganglia, the sympathetic and parasympathetic ganglia and plexuses, like the vast enteric nervous system that controls your gut. And they form the highly specialized hormone-producing cells of the suprarenal medulla, the adrenal medulla.
So what does this all mean? The neural crest is responsible for building a staggering range of systems, from our skin color to our adrenaline response to the bones in our face and the autonomous rhythm of our gut. It really is the tissue of complexity and integration.
It is. To summarize the full scope of this amazing versatility, you can group its derivatives by function.
Let's do it.
For sensory and autonomic systems, the sensory neurons of cranial nerves, V, 7, IX, and X, and all sympathetic and parasympathetic ganglia. For the endocrine system, the adrenal medulla cells that make adrenaline, and the C cells of the thyroid that make calcitonin.
What about structure?
For skeletal and connective tissues, the dentine, cartilage, and bones of the head and face. For pigmentation, the melanocytes. And for the cardiovascular system, the connective tissue of the heart, specifically for septation.
That list just underscores why understanding the EMT and migration process is absolutely vital in this chapter. A single defect in this process, a failure to detach, or a failure to follow the correct ECM highway, can compromise multiple, seemingly unrelated systems, like the heart and the skeleton, all at once.
It's a cascade of potential errors.
Now, the neural crest forms the scaffolding and many sensory components, but it doesn't build the cranial sensory system all by itself. It requires collaboration with specialized patches of surface ectoderm known as placodes.
That's right. It's a team effort.
What exactly is a placode, and why do they complement the neural crest?
Placodes are localized, transient thickenings of the surface ectoderm that are induced by the underlying neural tissues. They're essentially specialized sensory patches, and they contribute specifically to the cranial sensory structures that interact with the external environment.
So they work with the neural crest to form the cranial sensory ganglia.
Exactly. They collaborate.
Let's detail the key placodes and their unique sensory derivatives.
Okay. The nasal placodes differentiate into the olfactory epithelium and the olfactory receptor cells, giving us our sense of smell.
Okay.
The otic placode folds inward to form the otocyst, which gives rise to the specialized sensory structures for hearing and balance. That includes the vestibulocochlear nerve, cranial nerve 8th, and the spiral ganglion.
And there are others.
Yes, the pharyngeal arch placodes, which are also called epibranchial placodes. They contribute to the sensory ganglia of specific cranial nerves like 7, 9, X, and X, which are responsible for relaying taste and general sensation from the tongue and the throat.
So there's a clear division of labor here. The highly migratory neural crest forms the structural support cells, the myelin, the connective tissue around the nerves.
The scaffolding.
And the placos, which originate from the highly organized surface ectoderm, they form the actual primary sensory neurons themselves.
Exactly. The complex cranial sensory ganglia, like the trigeminal and vagal ganglia, are composite structures built by both. This intricate integration ensures that the cranial nervous system is fully formed and functional very early in development, allowing the embryo to begin sensing its rudimentary environment.
So if we synthesize the sheer density of information we've just covered, it's truly stunning. In the span of just a few developmental stages, stages 10 to 14, the embryo shifts from a flat, organized disc to a segmented, three-dimensional cylinder, a tube within a tube.
And by the end of that period, virtually every major cell lineage has committed to its adult identity. The blueprint is not just drawn, the foundation is poured.
The core lesson here, the key takeaway for you, the learner, is understanding that critical interaction between the two fundamental cell populations, the epithelial cells and the mesenchymal cells. This relationship defines the entire process of organogenesis.
It's a dance. Epithelial cells form the boundaries, the tubes, and the linings, the structured framework. Mesenchymal cells, through their flexibility, their migration, and their crucial production to the extracellular matrix, they form the muscle, bone, blood, and they provide the essential directional cues and scaffolding.
And if that dance is compromised?
If the EMT fails to release the neural crest, or if the notochord fails to provide that sonic hedgehog signal to the sclerotome, the entire body plan breaks down.
We have meticulously traced the specific derivatives of the three primary germ layers and the neural crest, referencing that detailed source map. We saw that cells from the paraxial mesenchyme can form both the skeleton of the vertebral column through the sclerotome and the dermis of the skin over your back.
Two very different tissues.
And yet, cells from the lateral plate mesenchyme can form both the connective tissue of your gut and the powerful involuntary heart muscle.
And that is an immense functional disparity from tissues that are immediate neighbors in the early embryo. They're separated only by a narrow space that completely dictates their signaling environment and their fate.
Which leaves us with our final provocative thought. We've described what these cells become in incredible detail, but the real mystery remains. What are the specific, minute molecular signals that determine these vastly different fates from such close origins?
The switches.
The switches. What precisely tells a cell that's released from the sclerotome that it must express the genes for bone, while its equally migratory sister cell, released from the nearby neural crest, must express the genes for a peripheral neuron?
The body plan is established, but the molecular signaling pathways that govern the commitment to these radically different roles, from bone to blood to brain, that represents the ultimate complexity underlying the simplicity of that early tube structure.
And that mastery of signaling is the true power and the true mystery of early organogenesis.